Okay, you should be taking notes on carbohydrates. You should have your graphic organizer in front of you. Let's get ready to go. First off, we want to talk about what elements are present in carbohydrates. And carbohydrates are really easy to recognize because they always have their elements in the same ratio. Carbohydrates always have carbon, water, and carbon and water. It's carbon, hydrogen, and oxygen. You can see CH2O. It's always in this same ratio, 1 carbon to 2 hydrogens to 1 oxygen. So you can tell very easily if you're looking at a carbohydrate. The monomer, or single unit of a carbohydrate, is called a monosaccharide. Mono means 1 and saccharide means sugar. The dimer, or the 2-unit carbohydrate, is called a disaccharide. Now remember from the earlier notes we took in class that the monomer, the single units, are going to bond together. When they bond together, water will be lost. So there's a water that's released, and that's called the condensation reaction to form that dimer. The polymer of the carbohydrate is called a polysaccharide. So mono 1, di 2, poly several or many sugars. There's 2 main functions of carbohydrates that we want you to know. The first one is that it gives some organisms structure. Examples of organisms that get structure from carbohydrates are arthropods and trees. Your bodies do not get their structure from carbohydrates. You get your structure from your bones, your skeleton, and your muscles that hold you upright. But molecules like glycogen and arthropods is what give them that tough outer shell, and that's a carbohydrate. Plants have cellulose. That's what allows trees to grow so tall and strong, because neither insects or arthropods or plants have bones. So they need to use the carbohydrates as a form of structure. Another function of carbohydrates is energy, and this one does apply to you. Carbohydrates are an excellent energy source. It has to do with the bonding of the molecules, and the more carbons you have bonded to hydrogens, the more energy you're going to have in your molecule. So there's these 3 major groups of there's these 3 major groups of carbohydrates that we want to be aware of: the monomers, the dimers, and the polymers. Let's talk about those, and let's write down some examples. So the monosaccharides and the monomers are some things you've probably heard of before. The examples that you want to be aware of are glucose, fructose, and galactose. Now these are single rings of sugar, the smallest sugar you can have, and they're going to have 6 carbons, 12 hydrogens, and 6 oxygens. Notice that ratio we were talking about before, CH2O. You see it here, 1 carbon for every 2 hydrogens for every 1 oxygen. Monosaccharides look something like this. They're a single ring-shaped structure. You can see that ring in the middle. And they're just a monomer. They're just a single unit. All 3 of the monomers of monosaccharides, the glucose, the fructose, and the galactose, they're all considered to be isomers of each other. This is also, in the picture in part B, is also a monosaccharide. This is the same general structure, but if you look very closely, there'll be a slight molecular difference. They're both C6H12O6, but they're just put together a little bit differently, so they become slightly different molecules. But they're both still small, sweet, sugary carbohydrate molecules. Dimers are a little larger, so this picture down here, C, is pointing at a dimer because I can see the 2 rings in the picture. They form during that condensation reaction, so you're going to have 12 carbons in this sugar. The actual formula is C12H22O11, which isn't our perfect formula of CH2O. But if you think about it, if I have C6H12O6 and I add that to C6H12O6, but I take out a water molecule, I'm going to have C12. Should have been H24, but I have to subtract these 2, so it's H22. and should have been O12, but I have to subtract the oxygen here, so it's O11. So that ratio will change slightly when we have that condensation reaction happening. The examples of dimers are also some things you might be familiar with, especially if you have done any cooking or baking. The dimers, the 2-unit sugars, are sucrose, maltose, and lactose. So still small molecules, but molecules that taste sweet in our mouths. The third major group of carbohydrates are polysaccharides. These are the polymers. These can grow longer and longer. So here I see 1, 2, 3 sugars in this molecule. Polysaccharides are more complex, and again, they can be from 3 to infinitely long numbers of those monomers all linked together. And the example that I would like you to know, the example I would like you to be aware of is glycogen in the animals and cellulose in the plants, those molecules that give the structure to the carbohydrates that we were just talking about on the last slide. So imagine you were sitting in your desk in the classroom and you were tasting the different solutions, the monosaccharides, the disaccharides, and the polysaccharides. You would be able to very easily tell the difference between the 3 solutions just based on how sweet they were. The sweetest solution should be the solution that had the smallest molecules. They're immediately picked up by your taste buds. So those would be the monosaccharides. The disaccharides, a little larger, are still sweet, but just not quite as sweet as the monosaccharides. And polysaccharides are starches. Think about eating a cracker or a piece of bread. It's not going to be as sweet in your mouth. Okay? So those polysaccharides don't taste as sweet because they're just larger molecules that your taste bud doesn't receive as a sweet taste. If you want to try something, go into your kitchen and get a saltine cracker, chew it up and hold it in your mouth. If you hold it there long enough, you'll notice that the saliva in your mouth is going to start creating hydrolysis reactions and breaking apart the polysaccharides into monosaccharides. So that long, starchy piece of saltine in your mouth is going to start breaking apart. You're going to have hydrolysis reactions at all these bonds, and you're going to get separate monosaccharides, and that cracker will actually start to taste sweet. In the lab, in class tomorrow, you're going to run a test to see if there's sugar present, and you're going to run a test to see if there's starch present in a solution. The sugar test is called the Benedict's test, and to run the Benedict's test, you just add equal parts of your testing solution and the Benedict's solution and heat it for a few minutes. Any kind of bright color change, you can see here in the picture, a bright red, a bright orange, a bright green, indicates that there's sugar present. The Benedict starts off as a blue color, so you see that blue reference color on the side. If it stays blue, there is no color change, there is no sugar in the solution. For our lab, you'll be using test tubes as well. You probably want to use about 20 drops of the solution and about 20 drops of the Benedict's. The test for starch is even easier. The starch test is called the iodine test. Iodine, if you look at the test tube on the left down here, iodine starts off as a yellowish color in solution, and if it does come into contact with starch, it turns a dark black purple color. So if there's starch present, you'll see the solution turn to a deep, deep color. Because we don't need to heat this one up, it'll just happen at room temperature. We're not going to use the volume of a whole test tube. We're going to use a small white dish called a spot plate. Add equal amounts of the solution and the iodine in the spot plate, maybe 2 to 5 drops of each, and see if it turns black. If it does, you'll know that there's starch present.